Welcome back to the Medical Microbiology Practical. This is Lecture 6 at College of Pharmacy in Jihan University, Dohuk. I am Dr. Marwan Hussain, and I'm going to present the culture of E. coli, Escherichia coli, on EMB and the indole biochemical test. You know, the E. coli, Escherichia coli, is one member of the Enterobacteriaceae. It is gram-negative routes. And as E. coli, cause a lot of infections, of course, and especially in gastrointestinal infections. The selective media which used for E. coli is EMB. So this is iodine methylene blue, EMB. It's a complex, chemically selective and differential medium. So the contents of the media, they contains lactose, sucrose, and the dyes iodine and methylene blue. So the sugars are included to encourage the growth of enteric bacteria and to differentiate them based on the color reactions created this reaction when combined with the dyes. If you remember that we have lactose fermentation, non-lactose fermentation. So E. coli is the lactose, E. coli and Klebsiella lactose fermentation, and the others like Salmonella and Shigella non-lactose fermentation. What are the purpose? What is the purpose of the dye in this media? The first one, this dye, like iodine and methylene blue, inhibit the growth of gram-positive organism, so no growth of gram-positive on this media. And these dyes react with the lactose fermenters. So in the acidic environment, the color will change, turn to the dark purple or black. This dark growth is typically of E. coli, usually and appeared as green metallic sheen. Green metallic sheen is the present or this is identified as it is E. coli colony on this media. The other bacteria of Enterobacteriaceae or other less aggressive lactose fermenters, such as Enterobacter Klebsiella, produce colonies, and the color of colonies is from pink to dark purple on this media. And of course, the non-lactose fermenters, null fermenters and sucrose fermenters retain this normal color or take the coloration of the media. Normal color or no color, on the media. So according to these colors and the reaction, we differentiate among the E. coli and other members of Enterobacteriaceae. So as you have seen in this images, this media, this is green metallic sheen. Green metallic sheen, this is E. It means that it is E. coli. Again, this is E. coli. This is pink, like Salmonella, Pseudomonas aeruginosa, And again, this is pink. This is a light pink, So, but Staph aureus as one of the gram-positive, no growth of staph of gram-positive. This is the colonies of Proteus on EMB, like it is like the waves of the sea. So what about the other test, which is indole test? So this test is we depend on this test, and we study and identify by this test the ability of certain bacteria to decompose, to break down the amino acid tryptophan to indole. Break down the amino acid tryptophan to indole, which carried out by some bacteria. So this, a tryptophan, when accumulated in the media, which produced by, of course, in the media which we have inoculate, and then if I use the reagent then I can identify and I can check the indole test, of course. Indole production test is important in the identification of enterobacteria, especially E. coli. So just only E. coli is positive and the other members of Enterobacteriaceae are negative. Of course, this chemically, or this reaction is performed by a chain of number of intracellular enzymes, a number of different enzymes produced, of course, by bacteria. So a system, generally, these enzymes referred as tryptophanase, because these enzymes break down the amino acid tryptophan, so it is called tryptophanase. So this test is a part of the IMVIC procedures. IMVIC procedures, it is indole, methyl red, Vogt-Broschkeier test. IMVIC, so a group of reactions, a group of tests. One of them is indole. So to check and differentiate E. coli from the others, or which bacteria are positive and which are negative among Enterobacteriaceae. As I said, the main principle of indole test. So tryptophan is an amino acid that can deamination and hydrolysis by bacteria that express and have this tryptophanase enzyme. So the indole is generated by reductive deamination from tryptophan to intermediate molecule indole pyruvic acid. This is explained in the metabolic procedures or styles, and tryptophanase catalyzes the deamination reaction. So when indole is combined with Kovacs reagent, this is the reagent which we use it when it is combined with Kovacs reagent, and this reagent contains the hydrochloric acid and the other chemicals and complex, of course, the solutions were turned from yellow to cherry red, because amyl or amyl alcohol is not water soluble. So from yellow to cherry red. And you will do this test by hand, of course, in the lab. So we will do together this test in the lab. So this reagent is the main reagent used for indole test. How can I do this test? Look, it's very important to inoculate the E. coli on the nutrient broth in the tube the day before the test. And after 24 hours in the 37 centigrade in the incubation. And then, so in this media nutrient broth, we have the E. coli inoculated in the media. And if I use the COVAX reagent, this is, as you have seen, this is a tube which contains nutrient broth, and I do inoculation of E. coli in this media the day before the test. And after 24 hours, I can add 0.5 milliliter of the COVAX reagent on this media, which inoculated previously by E. coli. And then the color, or the cherry red or ring, will appear on this media, on the surface of the or the top of the media in the tube. So this is positive, and this is negative. Why it appears? As I explained, so indole in this media, which produced by E. coli, when combined with and react with the COVAX reagent, so will give me this ring and this color from yellow to red cherry, and this is positive, of course. So E. coli, Escherichia coli, is positive, and the others negative, others members of Enterobacteriaceae. So by this test, I can do more identification, more deep, additional identification of E. coli, not only on the media. So this is additional reaction, additional identification of Enterobacteriaceae, E. coli and member of Enterobacteriaceae. This is our practical test. So by to do the inoculation, you will have, of course, we're going to do together in a group 2 media of EMB, and you will inoculate by the loop on the media, E. coli and other bacteria of Enterobacteriaceae like Klebsiella, Proteus. And the next day you will observe and check the colonies, and do the test, indole test as well, and then record your results on the data sheet. Thank you for your listening, and this is our practical session today.